All right, it's my pleasure to introduce Chris Knepp. He's currently finishing his 4th year as a PhD student in the Aquatic Animal Health Laboratory at Michigan State University and is pursuing a degree in fisheries and wildlife with a specialization in fish and wildlife disease ecology and conservation medicine. He has a bachelor's degree in zoology and a master's degree in fisheries and wildlife, both of which he obtained from Michigan State. His current research is focused on understanding Flavobacterium psychrophyllum genetic diversity as it relates to bacterial cold water disease ecology and management. And we've been following this work over the last few summers, so excited to see where you're at today, Chris.
All right, thank you, Dr. Phelps and the other organizers for providing us with another year to share our research findings. My presentation today covers a series of experiments that culminated in the development of 2 new culture media for the bacterial fish pathogen Flavobacterium psychrophyllum, which you can appreciate in the background of my slide. Before I begin, I'd like to acknowledge my co-authors, Drs. Call, Soto, and Locke, for their contributions to this work. So before getting into the details of this study, I'd like to mention an important trend in global fish production. So the orange portion of this figure shows you that we have historically produced fish for human consumption or other products through capture fisheries. But as you can see in blue, producing fish through aquaculture has been growing tremendously and is now the primary way in which we produce fish for human consumption. I constructed this figure based on data from the USDA, and it illustrates the impact of disease on US trout production. I know there's a bit going on here, but you can see we have millions of trout along the y-axis and years along the x-axis. What I'd like you to focus on is the red line, which shows the percent of fish lost to disease. So as you can see, this figure shows us that disease continues to be a significant impediment to the success of trout aquaculture. And what this figure doesn't show you is that we're losing fish other than trout to disease as well. And so the number of losses are even greater than what you see here. One of the most well-known and damaging bacterial fish pathogens responsible for these losses is Flavobacterium psychrophyllum, which causes bacterial cold water disease and primarily affects cold water fish species such as salmon and trout, not only in the United States, but globally. And as you can see in this image, psychrophyllum is exceptionally damaging to its host, and in severe instances of disease, losses can exceed 70%. So how do we diagnose a fish with bacterial cold water disease? Well, as you all know, there are a variety of ways, but the gold standard method is by recovering psychrophyllum via culture. The way we do this is by taking a sample from various lesions or tissues, in this case, a severe ulcer on the caudal peduncle, and culturing it on an appropriate medium. So now, best case scenario is that psychrophyllum will grow happily all over the medium, but this isn't always the case. Sometimes even on the exact same medium, psychrophyllum will only grow in association with host tissue, which may mean the medium is lacking components psychrophyllum needs to grow. Equally as challenging, sometimes psychrophyllum may not grow at all despite these significant active lesions and the detection of relatively high loads of psychrophyllum using other methods. So this outcome in turn then not only prevents the timely diagnosis of bacterial cold water disease, but can also lead to misdiagnosis and or prevent us from completing additional assays such as antibiotic sensitivity testing, which is necessary for recommending an appropriate treatment to prevent additional losses. So why could we be seeing these differences in psychrophyllum growth on routinely employed culture media? Well, it may be a result of the overwhelming amount of genetic diversity present within this species. So this figure is from a recent publication of ours and is an eBurst diagram of all currently recognized variants of psychrophyllum worldwide. Variants that are highlighted in red are present in the US. And of the more than 100 variants in the US, only 7 have been reported outside of the US. So most of the psychrophyllum variants here are different from the ones found elsewhere in the world. And so this is important to consider when we're developing new diagnostic tools such as culture media, because we should be incorporating these specific variants into our experiments. So psychrophyllum is a fastidious species. It can be difficult to culture, which is why so many different culture media have been developed for it. That being said, tryptone yeast extract salts agar or TIES, which was developed by Dr. Richard Holt in the late 80s, is the most widely used media type for growing Psychrophyllum today and is even recommended in the Blue Book by our section for detecting Psychrophyllum. However, nobody has ever verified that ties supports the growth of all variants equally. And then looking into the literature at the components of these other media types that were typically reported to improve Psychrophyllum's growth over ties, you'll find conflicting results where sometimes specific components improved growth, but in other studies using different isolates, growth was the same or worse. And so at the time these studies were completed, multi-locus sequence typing hadn't yet been applied to this species. And so these studies couldn't fully appreciate the wide diversity of Psychrophyllum variants that we now know exist. And so they were only using one or two to a handful of isolates. So because of this and the growth disparities we've witnessed in our lab, I designed a series of experiments aimed at creating a new and improved culture medium for the timely diagnosis of all currently recognized Psychrophyllum variants in the US. So the main question we were trying to answer in this study was, could we develop a culture medium that would improve the growth and recovery of most of the currently recognized US Phylobacterium Psychrophyllum variants? To answer this question, I came up with 4 objectives, the first of which was to compare Psychrophyllum growth on 3 different media types. The media type that recovered the most colonies for the greatest number of isolates would then serve as the basal or starting medium that would be improved upon in objective 2. For this experiment, I strategically selected 165 Psychrophyllum isolates that belonged to more than 105 of the 111 genetic variants in the US, which I highlighted in red. The selected isolates were geographically and temporally diverse, being recovered from 19 US states, the Canadian province, British Columbia, and the countries of Denmark and Chile over a period of nearly 40 years. And in addition, these isolates were also recovered from a range of fish hosts, including 12 different species. So for this experiment, I began by reviving each of these isolates on a medium that I devised and called neutral agar, because its components and their concentrations did not favor any of the 3 selected media types, which I'll talk about on the next slide. So next, I inoculated each isolate into broth and incubated them for the appropriate amount of time. After that, I harvested the cells and adjusted them to a standardized optical density. Then I performed serial dilutions and inoculated each isolate onto each of the 3 basal media so I could perform plate counts. The 3 basal media selected for this experiment were ties, which, as I mentioned, is a widely recommended and used media type for culturing Psychrophyllum, and Opplinger and Wagner, and enriched Anneker and Ortal medium with activated charcoal and aromatic compounds, which were both reported to improve Psychrophyllum's growth in comparison to other media, including ties. So these 3 pie charts summarize the overall results of this experiment. To reach this point, what I did was run an ANOVA for each isolate, where I looked for an overall significant difference in growth among the media types. If there was a significant difference, I made pairwise comparisons of the growth between the media types to identify the outcome. Those outcomes are what I'm summarizing with these pie charts. So the main thing to take away from these charts is that most isolates grew best on ties, and that more than 95% of the time, isolates grew best on ties, or ties performed as well as the other 2 media types. But it is also worth mentioning that some isolates grew better on OW or EAOCA and may have had different nutritional needs. But because ties had the most growth for the greatest number of isolates, it was chosen as the basal medium. So now that we had our best basal medium, the next step was to improve it. The way I did this was by using a Plackett-Berman experimental design, which is a convenient way of screening a large number of factors, in this case, medium components at multiple levels or concentrations. So I tested 11 different medium components at 2 different concentrations. I included the 4 medium components present in ties, which was the best basal medium. Looking into whole genome analyses of Psychrophyllum and its metabolism, I found that proteins and amino acid source are Psychrophyllum's primary energy source. So I included a number of components on that front. I also included rainbow trout blood, which I obtained from some of our rainbow trout that are housed in our quarantine facility, and it's a source of multiple host factors, including iron. And then for the concentration of each component, the high value is 50% higher than what's been reported in the literature for cycrophyllum media, and the low value is 10 times lower than that. So this way I was hopefully capturing the potentially wide diversity of nutritional needs of the different variants. And then so for this experiment, I used the same methods as I mentioned previously and selected cycrophyllum isolates that are representative of the variants in the US. Now getting into the results of this experiment. So this figure is showing you each of the 11 different medium components and the effect they had on cycrophyllum growth at the low and high concentrations. So the y-axis shows growth in mean log colony forming units, and the x-axis shows the low and high concentration for each specific component. You'll notice that 5 of these medium components are in red. This signifies that they had a significant effect on cycrophyllum's growth. So for example, cycrophyllum growth increased significantly from the low to high concentration for calcium chloride and magnesium sulfate, whereas an increase in concentration for caseamino acids and tryptose had the opposite effect. And then so with that in mind, these arrows show what concentrations were predicted to be optimal for the final medium. So based on those results, I produced 2 final media types to test, which I've been calling FPMA and FPMB. FPMB contains all the components at the concentrations I showed in the previous figure, while FPMA omits the statistically significant medium components that were suggested to be incorporated at the lower concentration. And then because rainbow trout blood did not significantly affect growth and is difficult to acquire, it was omitted from both new media types. Now that we had our new media types, it was time to compare them to our best basal medium ties using the same methods as before and 165 genetically diverse cycrophyllum isolates. So getting into the results of this final experiment, this figure is showing you the percent change in growth for 165 isolates when we compare FPMA to ties. So on the y-axis, we have the number of isolates, and across the x-axis, we have the percent change in growth. You'll notice on the x-axis, I included a red arrow that points to the left of 0 and a green arrow that points to the right of 0. The red arrow is showing you that 23 isolates had more growth on ties than FPMA. Between the 2 arrows, we have 8 isolates that grew equally on FPMA and ties. And to the right of 0, we have the 134 isolates that had more growth on FPMA than ties. So as you can see, FPMA improved the growth of most isolates, some of which by several hundred or 1,000%. And this figure is similar to the last, but instead compares FPMB to ties. So results were comparable to FPMA and ties, where some isolates grew better on ties or growth on FPMB and ties was equal. But for most isolates, growth was best on FPMB. And again, for some isolates, growth was improved by several hundred or 1,000%. So when we compile the information from the previous 2 figures together, we find that FPMA significantly improves cycrophyllum's growth over ties by almost 150% on average. And likewise, FPMB also significantly improves cycrophyllum's growth over ties. And lastly, FPMA achieved slightly better growth than FPMA or FPMB, but it was not a significant increase. Okay, now that we established that these new media types significantly improve cycrophyllum's growth in the laboratory, we wanted to compare their diagnostic sensitivity to ties in the field. To do this, we used FPMA, FPMB, and ties during our routine health inspection of 4 different salmonid species, including Atlantic salmon, Chinook salmon, coho salmon, and rainbow trout at 4 different spawning sites in Michigan. So for each species, I cultured the kidney of 60 spawning age fish onto each of the 3 media types and then incubated those at our lab for 7 days at 15 degrees Celsius. Now getting into the results, first I'd just like to orient you to what you're seeing here. So across the bottom are our surveillance locations, the fish species that were sampled there, as well as the media types that were used. the bars illustrate how many fish we recovered cycrophyllum from out of the 60 sampled at each site. And what you can appreciate here with these green lines is that on 4 out of 5 sampling occasions, both new media types were more sensitive than ties, resulting in an increase in detected prevalence by 6 to 13%. When we look at Swan River weir, where cycrophyllum prevalence was quite low, only FPMB detected cycrophyllum and from one fish. But so taken together on every sampling occasion, at least one of these new media types outperformed ties. So in addition to using the new media types for surveillance of cycrophyllum and apparently healthy spawning age salmonids, we also use these new media types and ties during 2 different mortality events involving juvenile Atlantic salmon, which were both being raised at 2 different Michigan State fish hatcheries. Here I'm showing you representative images of the Atlantic salmon that were submitted to us from both hatcheries. As you can see, fish had significant hemorrhaging primarily at or near the base of the pectoral fins, concurrent with gross evidence of an oomycete infestation. In a subset of fish, we also noted significant dorsal fin erosion where the fin rays were visible, which you can appreciate here in this image in the lower right. And this chart illustrates the prevalence of cycrophyllum recovered from the fins of the Atlantic salmon submitted to us from the 2 hatcheries on FPMA and ties. The other new media type was not used in these cases. So starting with the fish from hatchery A, you can see that the new media and ties were equally sensitive, recovering cycrophyllum from 50% of the fish. However, when looking at fish from hatchery B, ties did not recover cycrophyllum despite being prevalent at 25% according to FPMA. So this chart is like the previous one, but shows the prevalence of cycrophyllum in kidney tissue. As you can see, only FPMA was sensitive enough to recover cycrophyllum from the fish submitted to us from both hatcheries. And something I want to highlight is that ties did not recover cycrophyllum from the fins or the kidneys of fish submitted to us by hatchery B. And so without this new medium, we would not have recovered cycrophyllum and we would not have had the isolates in hand to perform antibiotic sensitivity testing to guide the optimal treatment recommendation. So since the time I submitted my abstract and now, we had a very recent mortality event at one of our Michigan State fish hatcheries involving rainbow trout fry. So these fry presented with disease signs very characteristic of rainbow trout fry syndrome, as you can see in these images, included severe cranial swelling and exophthalmia, as well as ulcerations on the trunk. So it was a bit last minute for this presentation, but I wanted to include some of the culture results for you all since I had taken the opportunity to again compare FPMA to ties. So what we saw in terms of overall prevalence was that both media types were able to recover cycrophyllum from the kidney and brains of 90% of the fish. But when we compare the number of colonies recovered between the 2 media types for a given fish, we see that recovery was substantially increased on the new media type, which fits with what we were seeing in the lab with my previous experiments. And so again, like with the Atlantic salmon, this new media type appears to be very useful for the recovery of cycrophyllum and the diagnosis of cold water disease. Okay, before getting into my overall conclusion, some of you may be wondering what about the cost. And so this table shows you the cost per plate for the 2 new media types and ties for our lab. You can see that the cost of FPMA and ties is very similar. FPMA is only 2 cents more expensive than ties, while FPMB, which was not significantly better than FPMA and has more ingredients, is a bit more costly. And so our lab has chosen to deploy FPMA over FPMB. So to summarize, I started with several observations, including that there was a disparity in the literature concerning which media type grows cycrophyllum best. I've also seen firsthand that some isolates are more difficult to recover from tissues independent of bacterial load. As a result, I started comparing cycrophyllum growth on several basal media and then was able to significantly improve upon a media type that has served us well for decades using an approach that hasn't been applied to cycrophyllum. And now we have two new media types that support numerous genetically diverse variants found across the US, offer significantly improved growth over ties in the laboratory, which will enhance cold water disease research, and have improved our ability to recover cycrophyllum from mortality events and during routine surveillance. And so these two new media types appear to be very promising on a number of fronts. So I'd like to acknowledge the USDA for funding this research, as well as the Howland Gene Glasson Conservation Medicine Fellowship and the Schrems West Michigan Trout Unlimited Fellowship, and my advisor, Dr. Thomas Locke. And I'd be happy to take any questions.
Great, thank you very much, Chris. Excellent talk there. Folks, feel free to drop a question into the chat box or unmute yourself. And to get things going, we have one here from Dave Groman. He's wondering if using explanted tissues was examined to determine if isolation is improved.
Explanted tissues?
Yeah.
I guess I'm not.
Yeah, Dave, if you can unmute yourself and clarify, that would be helpful.
Yeah, can you hear me?
Yep.
Yeah, typically in our lab, we will not take a swab or a sterile loop from a fish, but to improve isolation, we'll chop off a piece of the tissue. We push it into the typical media that was used in most labs. And then as the bacteria spreads out from that, we can pick out the pigmenting filamentous bacteria over other contaminants and then subculture that. And once we get a subculture of that, we put it into the mass spec. So I'm just wondering if you had attempted to see whether that would improve your isolation between the different media.
Yeah, so if I understand, you're saying you kind of stab the auger with the tissue and then allow the growth to happen around that.
Yes, both with gills and fins and body wall.
Gotcha. Yeah, so for these, we're using calibrated loops and then taking, a sample, in this case for, this large cell monads, it would be, 10 microliters first, like those Atlantic salmon would be one, and then those are streaked directly onto the plates. And yeah, I guess I haven't encountered really any issues with that. And I guess your question also kind of gets at, you're talking being able to pull out kind of the bacteria that you want to be able to pull out. So it's kind of getting at like the specificity. So in these natural mortalities or surveillance, I haven't performed any specificity experiments yet in the lab, but from what I've seen in the field, the new media seems to be a bit more specific than ties does in general for flavobacteria, and in terms of like other non-flavo, I guess, contaminants in this case. And we've had success using neomycin and tobramycin in the media.
Any other questions out there?
Hi, Chris. This is Charlie Giesiger. Great talk. I wondered if you've seen whether the new media for making like bacterial suspensions for susceptibility testing, whether you've seen compared live cells versus dead cells. I know, in making consistent suspensions, Sacrophyllum often, dies in media, and it makes it hard to make consistent suspensions for susceptibility testing. So have you looked at, whether your new media, the cells tend to die less than maybe compared to the TYES?
Yeah, so yeah, you're talking like growth in the broth comparing between ties and FPMA. So I haven't done any studies. I know some are done out there in media comparisons for other species where you would look at turbidity and optical density over time. I haven't performed any of those studies. I have been using the broth version of this, so just same ingredients, no agar. I haven't noticed any detriment to the growth or survival of Cycrophyllum over time compared to ties. It's actually now the go-to that I use over ties. So I haven't noticed any problems.
Great. I had another question also. Have you looked at your media, how with other closely related species like Branchiophyllum or different Chrysobacterium?
No, not yet. So the specificity testing is something that I do plan on doing, so that's something that we can look into and compare it to ties, because that's another thing that I don't think has been done for ties either. So it will be interesting to see. But no, I haven't tackled that just yet.
Great, thank you.
Yep.
Hey, Chris, this is Ron Miller from FDA. Thanks for the talk. It was really thorough and well thought out project. Just wanted to provide a thank you comment related to the confirmation results that showed the positive impact of the divalent cations, the calcium and magnesium, on the overall growth of the Psychrophyllum isolates. That's one of in our CLSI recommendations for susceptibility testing. We do recommend using cation adjusted Mueller-Hinton media for that reason. So your results kind of confirm our recommendations. So that's very helpful. Also, it was also helpful that you showed, correct me if I'm wrong, that the fetal bovine serum actually caused a reduction in growth when increased. So that, suggests, I think, that fetal bovine serum may not be that helpful for optimizing growth in vitro. So that's good to know for our purposes as well.
Yep. Yeah, and FBS is significantly more expensive than horse serum. So
Okay.
definitely a good thing. Didn't have to go that route.
Yeah, I think your project was really well thought out and thorough, and I think we're wrestling, some of us on our working group for susceptibility testing. We get inquiries from all over the world on how can I run disk diffusion on Flavorbacterium Psychrophyllum specifically, and we don't have any standardized methods for disk diffusion. We do for broth testing, but as you know, the Flavorbacteria on agar is challenging for a sensitive assay like susceptibility testing, where you need to have quality control and things like that to pass your test. So your overall project design could lend itself nicely to us, eventually optimizing the testing media. So I just wanted to put that out there. If you or anybody on the call is interested in that kind of work, we're always looking for folks interested in that type of investigation. Thank you.
Yeah, thanks for your comments.
Hi, Chris. Excellent talk. This is Haitham. Just a quick question. Have you noticed any difference in the colony morphology or characteristics of growth on your medium compared to ties? Stickiness to the agar, how spreading of the colonies, stuff like that.
Yeah, so differences in morphology and motility. It kind of varies depending on, I find that Psychrophyllum directly isolated from tissue appears differently than Psychrophyllum that's been handled in the laboratory for experiments. And then once you take like a primary culture from a fish and then subculture it, I find that the morphology kind of changes too. So talking about primary isolation, there aren't very dramatic differences in morphology. So that's been very helpful for me, because I was very accustomed to looking at Psychrophyllum growing on ties, and then a concern would be going to this new media type and having to relearn exactly how to differentiate it from other yellow pigmented bacteria. That hasn't really been so much of a problem. And then on subculture, it really kind of depends on the isolates. I would say that there probably is a bit more gliding motility on the new media than there was on ties, but largely very similar.
Okay, good. Thanks. And I've learned something new today when you said you isolated from the kidney on your media, but not on ties. We usually thought that, the challenging part is isolation from the external organs, from the skin and gills, because of the other competitive bacteria that takes over when you isolate. But, for you to isolate from the kidneys on your medium, but not on ties, this was something interesting to know today. Thank you so much.
Yep. Yeah, it was definitely one of the driving factors, especially it seems to be more difficult with the Atlantic salmon where you tend to get that saprolignia or, some sort of oomycete secondary infection. So that was one of the driving factors to get the new media, and kind of now we're working on, dialing in some antibiotics, and it seems to really help prevent the growth of, the non-targets. So
Yeah, great work, and thank you, and thanks to your co-authors.
yeah, thanks.
All right. Well, thank you very much, Chris. Great talk.